welcome everybody here from the Kansas City campus. And thank you very much, Dr. Wilson-Rawls, for joining us today. We're all excited to hear your presentation. We have a whole bunch of students here in the auditorium, as you can probably see. And I will turn it over to Dr. Staudinger.
Hello, colleagues. It's my distinct pleasure to welcome Jeanne Wilson-Rawls to the KCU Science Fridays seminar series. Dr. Wilson-Rawls received a Bachelor of Science in Biology from McMaster University, north of the border in Hamilton, Ontario, Canada. Subsequent to that, she received her PhD in cellular and molecular biology in Missouri at St. Louis University School of Medicine, obviously in St. Louis. I met Dr. Wilson-Rawls in Texas at the University of Texas Southwestern Medical Center, where she was a postdoctoral fellow with Dr. Ralph Arlinghouse for a year or two, and then came to join the lab of Dr. Eric Olson. And Jeanne and I had the experience of moving Dr. Olson's lab from M.D. Anderson Cancer Center in Houston to UT Southwestern Medical Center in Dallas. And that was an adventure. Dr. Wilson-Rawls and I became colleagues in around 95. And I must say that her mentorship and her teaching of various techniques was actually instrumental in my development as a functional graduate student and scientist. After leaving Dr. Eric Olson's lab, Jeanne joined the faculty at Arizona State University in 1997, where she's been there for, it appears, 24 years, if I'm doing my math correctly. She's currently an associate professor of genomics, evolution, and bioinformatics in the School of Life Sciences at Arizona State University. Dr. Wilson-Rawls has numerous affiliations and honors. I'll just mention two. She's the director of the graduate program of molecular and cellular biology, an interdisciplinary program. She's also co-chair of the American Heart Association of Basic Cell and Genetics panel. She's received numerous awards, two of which I'll mention: the Flynn Distinguished Researcher of the Southwest Affiliate of the American Heart Association, as well as she was nominated for the Dean's Distinguished Teacher in the College of Liberal Arts and Sciences in Arizona State University. Numerous invited talks, well over 40 peer-reviewed publications, and book chapters, receiving funding from NIH, the Muscular Dystrophy Association, American Heart Association, as well as the Arizona State University Smithsonian Tropical Research Institute. So Jeanne, it's a pleasure to see you again. Take it away.
All right, well, thanks for the very kind invitation and the very nice introduction. And so I thought that, from my understanding, Jeff, that we had a very broad audience today, so I decided to take a dim sum approach, and I'm going to talk about a little about a couple of different projects that we have ongoing in the lab. The first one we'll talk about is regulation at the molecular level of satellite cell function during muscle regeneration. And then I will also talk about our studies of satellite cells that come from reptiles, in this case Anolis carolinensis, that can regenerate entire limbs. And the last piece that I'll talk about, and this might be more applicable to a med school audience, is about inflammation and regeneration in dystrophic muscle. So for those who are not muscle aficionados, there's a stem cell, and here's one right here on this muscle fiber, known as a satellite cell, which is a muscle progenitor. You know, you go to the gym and you work out and your muscles ache because you make lots of little micro tears. And this stretch response will actually activate these little cells. And then over a well-studied period of time, you can see that there is the changes in gene expression during first the expression of the muscle cells. They're gonna turn on MIF5 and MyoD. As repair continues and they have to make a decision, they start to turn on myogenin and MEF2, and then you get muscle structural proteins coming on. And this initial period is part of a Th1 sort of pro-inflammatory response, and as we move through time, by about day 6 or so, we're moving into an anti-inflammatory response. And by about 10 days, you'll have whole new muscle fibers and repaired fibers. And so we have this nice, well-studied model to use for studying stem cell commitment during regeneration in mammalian muscle. And so if we look at it here, here's our little cell. It gets activated. They divide a lot. They have to make a decision at this stage where they're expressing MyoD and MIF5. They're starting to turn off PAX7. They'll also maybe express a little bit of Syndecin-4, maybe. And some of these will be decide that they're going to be going back to the niche, and some will turn on myogenin and MEF2C, and they're going to fuse into repairing fibers, and they're going to make new fibers. And what people have found is that you see expression of Notch in these cells, and you see high levels of expression of NUM in these cells. And so the Notch signaling pathway has long been felt to be one of the big regulators of this commitment versus self-renewal decision. And we started looking at this because here's the standard version of this decision. Here's our muscle progenitor cell. If it expresses Notch in both daughter cells as it's dividing, you get 2 more progenitors. So we're making more cells. At that point that decision has to be made, they'll suddenly start to express NUM on one side of the cell during division. So this would be an asymmetric cell division. The cell that has NUM in it will become a myoblast and express myogenin. The knockouts, though, of NUM, embryonic lethal mutation, there's a gene that is nearly identical known as NUM-like, and it seems to be completely redundant to NUM. If you knock it out, you get a fertile, healthy mouse. And so we decided to look at this decision by knocking NUM and NUM-like out using conditional knockouts. And we wanted to see, could we disrupt this decision, and would we get something that kind of looked like a rhabdomyosarcoma? Because at the time that we started this project, we thought it makes sense. Notch is involved in many different tumors, as is NUM, so why not having a role in rhabdomyosarcoma, which, for those of you who may or may not know about this, rhabdos have not changed in outcome in about 50 years. We do know something about some of those that have translocations with the fork head genes, but otherwise we don't really understand why these cells stop differentiating and stay proliferative. So the first approach was to knock it out embryonically. So we used a PAX7 Cree with the NUM flocks in a Rosa YFP mouse, and we bred it onto a NUM-like null background. So you have no NUM-like, and you have this PAX7 Cree just knocking NUM out in developing cells that are going to become the satellite cells. And if we look here, we've stained for Syndecin-4 on these myofibers, and here's our PAX7 stain on myofibers. So with the YFP, so here are the cells that have recombined in the presence of our Cree. And what we did was we looked at those myofibers in culture, and we found that we don't get proliferation in the same way, so they have many fewer satellite cells on the fibers when we knock out NUM-like and NUM. Whereas if we have a heterozygous floxed NUM with NUM-like there, we have pretty normal proliferation of those satellite cells. So our average number of cells per fiber is fewer. And when we looked at damage, so we took muscle and we injected it with barium chloride, and we looked at the muscle, and you can see here is our numb, flox, numb-like, null background. And then here's the PAX7, numb-like flox, numb, null background. So this one is missing numb, this one still has numb. And at 4 days, they don't look terribly different, but at 14 days, you can see that we're getting, still have a lot of fibers with centralized nuclei, indicating they're still trying to repair themselves. But we also have increased fibrosis, this clear area here, and fatty infiltration, indicating that we're not getting good, efficient regeneration of this muscle. When we measured the cross-sectional area of those repairing fibers at both 5.5 days and 14 days, so 5.5 would be right around that switch from the inflammatory to pro-inflammatory, so you should have very small fibers that are trying to repair themselves. And at 14 days, we should have fully reformed myofibers. We can see that if we look at the PAX7 flox numb-like null, we have at 5.5 days, they have smaller fibers than something that has no numb-like but has numb. And at 14 days, they haven't increased in size. So they're still about, 400 microns squared. And here we have much bigger fibers in our other genotypes. So here's the presence of numb-like in the absence of numb, and then the presence of one copy of each numb-like looks pretty normal. So we have less cells, less cells, and we have impaired regeneration with the kind of fibrotic changes that we typically would see in a situation like Duchenne's muscular dystrophy, where you're getting fibrosis and fatty infiltration. But we knocked this out embryonically. And so one of the questions that we still had for ourselves was, did we actually impair the formation of proper satellite cells? So we took another approach, and we used the PAX7, we used the ER-Cre, bred it into the numb and numb-like floxed mice. And so in this case, you allow the animal to develop. They will develop normal satellite cells. And you can then use tamoxifen to knock the gene out selectively in the satellite cells. And so that's sort of a long process. You do a series of tamoxifen injections over at a 60-day-old mouse. You wait a month, basically, to let them recover, because they don't look so good after you treat them with tamoxifen. And so then we use cardiotoxin injection, and we harvested muscle at 7 days and 10 days. And we looked at what happened during repair. And this is Trichrone staining here. So here's our ER-Cre double floxed animal, and this is the damaged muscle at 10 days. And you can see there's a lot, this blue stain here. These are necrotic fibers. You can see that there are very small fibers interspersed in here with centralized nuclei, but there aren't very many of them. When we have numb floxed, but on a numb-like wild-type background, we still see a lot of necrotic fibers and very small fibers with centralized nuclei. We also see heavy infiltration of inflammatory cells. Whereas if we look at the ER-Cre on a wild-type numb, you see pretty normal looking muscle, centralized nuclei all repaired itself. With no Cre, you see something similar, although we did find this was interesting. Consistently, these animals showed a little bit more infiltration of the macrophages, and we're not really sure why. And here's the heterozygous animal, heterozygous on both loci, and it looks really normal. When we looked at cross-sectional areas at 10 days, we found that The only difference was our double-floxed pre-animals had much smaller cross-sectional areas of the regenerated fibers than any of our other genotypes. And so we've now knocked it out. Embryonically, we used an inducible pre to knock it out in an adult, and we found that muscle regeneration is impaired. So we were kind of surprised by this. We expected to see a little bit of impairment, but we expected to see actually increased division, so many more satellite cells, and we just didn't find them. And so we went ahead and said, okay, let's just take the satellite cells out and put them into culture in vitro and ask what's going on. And if you look at this graph here, this is a proliferation experiment where we've counted the cells every day for 6 days after seeding each well with 10,000 cells. And this line and this line are both num knockouts. One is heterozygous one has normal num-like and one has no num-like. And these are all the other control genotypes. And you can see that over time they grow very well, whereas these guys didn't grow at all. Well, there's two reasons why you might not see proliferation in such an experiment. One is the cell is actually not growing, and the other is that you actually have pretty normal proliferation in some cells, but you also have a lot of cell death. So we did assays for caspase, we did live dead assays, and what we found was sort of interesting. So the satellite cells were there, they were not dying, but they were also not proliferating, which made us think about quiescence. And so we wondered about things that are involved in quiescence. And we first said, okay, well, maybe they just are impaired in some fashion, that they can't differentiate. So we put them at fairly high density and then either ethanol treated, so this is our control for treating with tamoxifen, or we added 4-hydroxytamoxifen to the culture media and asked if they could differentiate. And you get pretty normal differentiation when you knock out both num and num-like, or not. And when you add tamoxifen, so now you've knocked these out in the cell, you do not get, you get these very skinny looking things with very few nuclei. They do not form these giant networks that are full of nuclei that you see when you don't add the tamoxifen. So clearly a lack of num was resulting in this hypoplastic differentiation and lack of proliferation. And we started looking at genes involved in both cell growth, cell cycle, the Notch pathway, and the myogenic regulatory genes. And the only thing that we found that was significantly different were both myostatin and P21. And we could find that there was just so much myostatin being made in these, this is a Western blot, in these cells compared to vehicle treated cells where we couldn't detect it at all, or just wild type satellite cells that were treated with tamoxifen. So it's a very large increase in the amount of myostatin and P21. And so this is really interesting. P21 has been shown to inhibit MyoD. It also inhibits cell cycle. And myostatin, you probably, if anyone knows anything about muscle, they know that myostatin is the brakes, basically. It turns muscle cell proliferation off, and it keeps muscles to a certain size. And I don't know if you've ever seen pictures of the Belgian blue cows, the super muscle cows. So they all have myostatin mutations in them. So we found this really interesting, that in the absence of num, that these two particular genes were highly upregulated in expression at both the mRNA and the protein level. So Where we expected to see a difference was that we expected to see a loss of self-renewal and perhaps an overgrowth of these cells without commitment. But it turns out that in the absence of NUM, these satellite cells simply don't want to grow. They don't really break quiescence for some reason. So they're stuck in this quiescent-like state. Currently, what we're doing is proteomics screens. We found a way, and for the students who were reading the paper, we found a way to take cells, put them on very soft hydrogels, and get active proliferating cells to step back in time and become what we're calling a Q alert stage. They're not completely quiescent, but they shut off myogenin, they re-express PAX7 and MIF5. And so we're taking these cells, immunoprecipitating NUM and doing screens, because what we're really interested in is why NUM has this previously unrecognized role in the actual activation of these satellite cells. No one has, and we went back and looked at some really old papers too, and it's very interesting that people who looked at very early cells found NUM expressed in this population, but really later, we only talk about it here in these activated cells. So we inadvertently found a new role for NUM, and we're actively trying to figure out what the targets of NUM are to understand how NUM has a role in breaking quiescence. And of course, this is an ongoing project. I think I forgot to mention who did this work. Okay, so this work was all done by two really talented grad students, Brian Barris and Rajni George. Brian is got a position on the East Coast where he's teaching, and Rajni is currently a postdoc in Tony Ferruli's lab at Indianapolis. One of the things that I and many, many others have asked is, can satellite cells be used therapeutically? If you and I have volumetric muscle loss from trauma or surgery, we can't really repair. We can repair the muscle that's left, but we can't remake whole new muscle groups. And so a lot of times people who have volumetric muscle loss are disabled, not because they've lost a limb, but because they've lost the ability to move the limb because they don't have muscle. And we decided to approach this from a slightly different angle than many other people and ask, how do animals that regenerate whole new muscle groups do it? And we looked at this little guy here. This is Anolis carolinensis, or the green anole. This is native to North America. And we started to look at how does tail regeneration proceed and what happens to the muscles. And this is the work of three very talented students. My own student, Joanna Pilate, Elizabeth Hutchins, who was in Kenra Kasumi's lab, and George Jordovic, who was in Josh Ho's lab at Victor Chang. And the first thing that we did was we started to look at how muscle develops in that regenerating tail. And what you can see here is this is a sagittal, frontal, I don't know, maybe sagittal section through very early tails. Are about 20 days post-autotomy. Lizards will do this thing where they'll just drop their tails and they'll go scurrying away. It's an anti-predation event, and it's really interesting. The vertebrae have fracture planes, and all of the blood vessels there have valves, so there's almost no blood loss, and it's designed for the animal to just have that tail drop off. And so you can make that happen by simply pinching the tail with your hand, and they'll just drop it. And then it will wiggle a lot. That's supposed to distract you as the predator, and the animal can scurry away. And so what we found is here's the old vertebrae, there's the bone, and right on top of the bone you can see this cartilage tube forms that extends down the length of the growing tail. Within it you get regeneration of the spinal cord and ependyma, and out here we have regeneration of connective tissue and muscle, and we see growth of nerves from above the break point down into the muscle. and we regenerate scales and chromogens. And when we just look here, we've stained in this section for myosin heavy chain so we could see where the muscle groups are. You can see that all the way along, we're getting the formation of these little muscle groups. Well, if you were taught the traditional epimorphic model of regeneration, there would be a region right here of undifferentiated cells that are highly proliferative. And that would be like in the limb development model, your outgrowing region of this appendage. And we just weren't seeing that. And we found that this was curious because, of course, we'd all been taught the same model. And we also find that we have single nucleated cells all interspersed within here, as well as very short fibers that are starting to join together all the way along this tail. So this was kind of a surprise to us. And so where are the regenerating cells coming from if we don't have that blastema at the outgrowing end, which is supposed to be this dedifferentiation of adult tissues and formation of this proliferative blastema? It's possible we're looking at some sort of stem cell mediated embryonic-like event, and maybe these stem cells are lineage restricted. And so here's the traditional model. Here's this blastema. And as it grows out, you start to get differentiation behind in the regenerating animal. So here's our amputation plane. There's the formation of the blastema. The whole thing grows out. And behind it, you're starting to get redifferentiation. Well, this really wasn't what we were seeing at all. And in fact, you know, this is seen in planaria. Planaria are awesome if you want to look at regeneration. You can cut them into a zillion pieces and they will just regenerate whole new little worms. And it's also seen in salamanders where you get this. But recent studies showed that not only are there lineage restricted cells within this blastema, so in other words, the cells remember where they came from, that the muscle cells in particular only made muscle in the regenerating limb of a salamander. So salamanders are these super regenerators like planaria. If you cut off a limb, they'll regenerate a faithful limb exactly the same. Lizards don't really do that. They regenerate a tail. It's not exactly the same as the old tail. And again, muscle really only becomes muscle, as it turns out. So we think that in the lizard tail, we're only getting this stem cell mediated type of regeneration, that we're not actually making this blastema. And so if we look at MCM2 expression in a regenerating lizard tail, MCM2 is a marker of proliferating nuclei. And you can see that here's developing muscle all through here, and they're all positive for MCM2. The ependymal cells in the middle of our cartilage tube are. All of the skin and some of the connective tissue. But what you don't see is you don't see a true blastema. So we started to ask questions about what these cells might be. And we decided to pull out muscle and look at whether they have satellite cells. So if you were doing this with mammalian cells, you'd probably use flow cytometry to isolate your satellite cells. There aren't really commercially available antibodies for lizard proteins, so we had to do some work at validating that we could find these cells. And so we took cells from muscle and we used the pre-plating technique with mammalian satellite cells. They'll stick down on a plate if you have a matrix there, but they won't if you don't. But other cells that come out of the muscle when you grind it up and make a single cell suspension will happily sit down. So we use a series of pre-plate events to take that floaty layer of satellite cells off and then finally put them on matrigels so they'll grow. We had a lot of fun trying to work this out. It turns out we have to grow them at 30 degrees Celsius. At 37, they all died. 35, they just grew mold. It was really awesome. So 30 degrees is the term. And when we grew them on cover slips and stained using the Anti-Mouse Pac7, we had already done RT-PCR to convince ourselves because we have the genome, and Kenro's lab generated probably 30 transcriptomes, one of which was from muscle. So we knew that Pac7 was there. And it turns out that lizard Pac7 can be lit up by the anti-mouse Pac7. And so here it is here. This is DAPI staining of the nuclei. Of course, Pac7 is a transcription factor, so it's also nuclear stained. So you can see similar pattern in both the lizard and the mouse cells, and with no primary on the lizard, we don't see stain. So that was really happy. We found cells that actually look like they might be satellite cells. However, considering that lizards can regenerate whole new muscles and they have new tendinous attachments that are not the same as the old tendinous attachments, we were wondering, are these really satellite cells or are they some other musculoskeletal mesenchymal type stem cell? So we did some transcriptome work and we compared the genes expressed in mouse Pac7 positive satellite cells and lizard Pac7 positive cells. And it's kind of hard to do this. So Josh and George and Kenro developed an algorithm to allow for this cross-species comparison of genes in transcriptomes. And what this does is that you take your data from each transcriptome and you rank based on the number of copies or times that you hit that gene. So you create this rank list. Then you take the cross-species rank lists and we use an algorithm to compare them. We fit these to a linear model using Spearman's correlation, and then we have a way to compare what genes are being expressed in mouse and lizard satellite cells, where we don't have to worry about, are we looking at the same gene? Is that really the same transcript? Because we're looking at them based on this ranking. And you can see here, this is the mouse lane and this is the lizard lane. Here we're looking at TGF-beta pathway genes. TGF-beta is really important in muscle, so we looked at that, and then just generalized musculoskeletal genes. And you can see that there's some strong similarities, but there's also some very big differences. And so one of the things that we found was, oh, I should back up a bit. One of the things that we wanted to do with this data set was start to ask questions, are there underlying specific differences in gene expression that would start to point us toward why it is that the same basic cell from lizards could regenerate whole muscles, but ours can't. And found some interesting things. The first thing that we wanted to do is we wanted to see how similar were they to satellite cells, and were there other cells that we might find that they're very similar to. So we looked at the ENCODE database, and we looked at all of the ENCODE transcriptomes, and we found that the two top hits when we compared these lizard cells were mouse satellite cells, limb embryonic cells, which are mostly muscle cells, human activated and human quiescent satellite cells was the other group. So we looked at transcriptomes from 90 and 136 species, and we came up with our top hits all being satellite cells or muscle. The other really interesting thing was we found that cartilage was within the top 15 most closely related transcriptomes, and we were really surprised by this. And we found that when we compared first the TGF-beta pathway, so TGF-beta is really important for cartilage, it's also really important for muscle. And you can see, so here the dark blue is a high level of expression, the dark red is low. And we see some strong differences with MSX2 and RNF 1655, but otherwise they look pretty similar. If we look at, these are muscle genes and other transcriptional regulators. So here's MEF2C, we're expressing a lot less MEF2C in the animal cells as compared to mouse satellite cells. We also express a lot more of these other muscle and early developmental genes in these cells. And when we looked here, we also found this was true down in this region here where we're looking at SMADs. These are other signaling molecules that, again, we have a lot less expressed in the animal cells in general than in the mouse. So for some reason, overall, we're not expressing sort of muscle-specific genes so strongly in the animal cells. And the genes with chondrogenesis are upranked as well in the animal. So we found that really interesting. So when we said, okay, fine, let's just look at the top 20% of the highest ranked genes based on the Go terms for muscle, cartilage, and tendon and compare them between the two populations. And what we found was that overall, mouse satellite cells express the most muscle-specific genes in that top 20% of genes. There was some cartilage genes, and there was some that overlap in function. But if we looked at lizard, in that top 20%, only 27 of the genes were muscle. But we have this increase in cartilage genes overall. And so this led us to ask, is part of the reason that those cells can form whole new muscle groups with tendons is that they are perhaps a little bit earlier in development. They're not truly a satellite cell, but they're a musculoskeletal progenitor cell. And so we went back and we looked just at the BMPs. So here's BMP2, 7, 5, 6, and 4. And satellite cells, if you take a mouse satellite cell and you give it exceptionally high concentrations of BMP2, 5, and 7, you can force those cells to become chondrogenic within culture. But we're talking super physiological doses, like on the order of 500 nanograms per mil. And what we wondered was, because we could see that there was increase, especially in BMP4 here, which is really important for development of cartilage. And then here you have almost none over here, BMP6, and we have limited amounts of 2, 5, and 7. We were like, well, I wonder what's going on. But this was sort of hopeful here. And so we took these and we said, we'll just put them at high density micromass, because if you want to grow cartilage, you need the cells to be at high density. And we'll put them in either regular growth medium or a chondrogenic diff medium, but we won't add BMP2, 5, or 7. And we'll look to see what happens. Can these cells actually become cartilage? And they're really under this, we're also going to look for the expression of things like SOX9 and glycosaminoglycans and other markers of cartilage development. So if we look at what happens during development to make skeletal muscle, you see PAX3 and 7, you see the notch num pathway and the myogenic regulatory factors are all coming in the mesenchymal cells. But if you're looking at cartilage, early on, you start to express SOX9. It's maintained as chondrogenic mesenchyme forms. In pre-chondrocytes, we also see SOX9. We also see expression of BMPs 2, 4, and 7. And we start to see expression of collagen 2A1. As this continues, it will switch to collagen 10 and it will start to express aggrecan and other glycosaminoglycans, whereas tendons express scleraxis, and then they express things like fibromodulin, biglycan, and corin. So we can look for evidence that we are picking one of these pathways by looking at what these cells express once we put them in culture. And the first thing that we noticed is just looking at them morphologically, we could see in chondrogenic medium that we started to see these nodules that sit up off of the monolayer, and you can actually pick them off with a pair of forceps. So they're hard and solid, and they started to become a little bit acellular. We don't see that with these cells in growth medium, and we don't see them in mouse cells that are in chondrogenic medium. And in fact, mouse satellite cells really want to become muscle. In growth medium, if you leave them too long, they start to fuse, and so you can see there's a couple of big fibers forming here. But in chondrogenic medium, where we actually should be inhibiting muscle formation, we are still seeing the formation of myotubes in the mouse satellite cells. So these cells from the lizard are inherently different in the sense that they respond to the chondrogenic growth medium and they will form these nodules. And so the next question that we wanted to assess was what's being expressed. And we found that we could not reliably get RNA out of these nodules, so we had to go a little bit earlier in time to harvest these cells and look at the RNA to see what's going on with gene expression. And we found that if we just put the cells in growth media versus diff media, and here the lizard are the light gray bars and mouse is the dark gray, we found that both started to drop the expression of PAX7, which makes sense because they're all starting a little bit to differentiate. And as you keep my satellite cells in culture, they start to lose PAX7 expression. But we found that both cases, PAX7 expression was higher in the lizard cells than in the mouse cells, which we found kind of interesting. MyoD, if we looked at growth medium versus diff medium, the lizard cells start to decrease the amount of MyoD that's expressed significantly, and correspondingly, we see this upregulation in MyoD. It's very low, though, still compared to day 0 in our mouse cells. RUNX2 and SOX9. RUNX2 is, along with SOX9, are expressed early in chondrogenesis. If we look at RUNX2 here in growth medium versus diff medium at day 7 in culture, our lizard cells upregulated, but even in diff medium, the mouse cells will start to upregulate it. So in response to just being in that environment that inhibits myogenesis, you start to see RUNX2 coming on in these cells. But when we looked at SOX9, which is really that central core regulator of chondrogenesis, you can see this massive upregulation in both growth medium and diff medium by the lizard cells, and almost no change in the mouse cells. So when we look at RNA, the lizard cells are definitely able to turn on that master regulator. Satellite cells from mouse will make a good go of trying to turn on RUNX2, but aren't able to turn on SOX9, and these cells also downregulate MyoD. When we look at other cartilage-specific genes in this experiment, and we can see, I just want you to look here and here. So we had shown you earlier that we could turn on SOX9, and we get some turn on RUNX2, but what's really interesting is without giving them BMP2 or BMP6, we can see that the lizard cells are turning these on. And the really interesting thing here is we chose BMP6 because it has nothing to do with cartilage development. It's actually involved in iron metabolism, so we know that it's expressed fairly well in most satellite cells. And so you can see that here. But as they diff, they start to turn it off. Our lizard cells do increase it in diff, but not in pulp growth situations. But again, it's iron metabolism. We're not sure how much iron metabolism cartilage really needs. However, BMP2, which is really important for formation of cartilage, is highly upregulated. And here's our extracellular matrix genes. And again, we're looking at high levels of expression in the lizard cells in differentiation conditions. So when we looked, here's our cells. We decided, well, we should really ask what's in those nodules since we couldn't get RNA. And so we stained for collagen 2A1 in growth and diff conditions. And this is the mouse cells up here, and here are the lizards cells. And you can see once again, we have no real nodules in the mouse cells, but the lizard cells, even in the growth media, we're starting to get these little nodules forming that are positive for collagen 2A1. And under diff conditions, we get these very large collagen positive nodules. If we just took a section through a regenerating tail, here's the collagen 2A1 antibody in that cartilage tube when we've done immunofluorescence, and here's the no primary control. And you can see that this lining lights up really well on the ependymal tube. So lizard satellite cells, unlike mouse satellite cells, will turn on the differentiation program of cartilage. They express SOX9 and the BMPs, and they can form these cartilaginous nodules. So they clearly have expanded musculoskeletal ability. And one of the things that we would like to do is we would like to use geogenomic kind of approaches where you can, there's a company out now that has, you can take sections and you can superimpose different regions using a computer. It will go in and create transcriptomes from different regions of a section on a slide. So you can start to ask questions about where are these cells? What are these cells? What are they expressing? So that's where we'd like to go with this project. And when we tried to look at how many nodules were seen, because these kind of look a bit like they're trying to form nodules, we let them go to 10 days in either diff or growth medium, and we could find that there were some small nodules that were being made in mouse, but you can see the difference here. And the surface area is quite different too. So this project, we started asking questions about why we can't use PAX7 positive satellite cells in therapeutics by asking about the animals that can regenerate appendages. We found that the satellite cells have expanded musculoskeletal ability. We're still not really sure. We're still looking at the transcriptome data for what might be the big regulators of this difference. And we're moving forward with trying to find out where these stem cells are in that regenerating tail and what do they really express. So my last story is done in collaboration with Alexandra Lucas and Alan Rawls, and it has to do with inflammation and regeneration in dystrophic muscle. So dystrophin is the largest gene in the human genome, and it's right here, part of this whole complex that's found at the plasma membrane of the muscle fiber. And basically, you can think about it as a giant shock absorber. So it helps to buffer that membrane with contraction. In people and patients without dystrophin, everyday use of your muscles and contraction can cause all kinds of damage. And unlike this story here, where this is an acute muscle model where we've caused the damage, we see this initial inflammatory response, and we have upregulation of the regulatory genes, followed by an anti-inflammatory response with regeneration. In Duchenne's muscular dystrophy, you have this constant damage being caused by everyday use of the muscles because of the lack of dystrophin. And oh my God, stop. And this causes this whole inflammatory environment to be dysregulated. And so you go from this acute environment to a chronic inflammatory environment. Muscular dystrophy is quite debilitating. Most Duchenne's kids are diagnosed by the age of 3. They're no longer ambulatory in their tweens, and many of them die in their 20s because of heart and lung issues. And so it's a big problem. So if we look at immune mediators in this whole process, that within hours of trauma, we find there's this whole cellular response. And we can track it by looking at things like INOS, tumor necrosis factor, chemokines such as CCL2, IL-10, and IGF-1. And so we can look at both the soluble mediators, and you can see they have also a time frame similar to the rapid inflammatory response and the later repair response. We can also look at it based on infiltrating immune effector cells, such as neutrophils. M1 macrophages come up first, which makes sense. They're pro-inflammatory, followed by the many types of M2 macrophages, which are anti-inflammatory. We have T regulatory cells. You can see these are some CD8 positive T cells. So they change as you do not want to do that. It all follows this nice pathway. What you find in Duchenne's muscle is we're sort of stuck here. We never get out here. And so there is increased immune effector cells, there's increased fibrotic damage, there's increased fatty replacement, and this all leads to loss of muscle tone and ability to functionally use our muscles. So we've been using a virally derived serpin called SERP1, which activates the UPAR pathway. And serpins are known to have roles in inflammation. And the first question we asked, Alex wanted to know if we took this virally produced SERP1, and it's slightly different than the normal serpins, and I'm not allowed to tell you why because she's got a patent out, is that would it work in improving the timing of acute muscle injury? And what we found was that there's a slight shift to newer fibers and bigger fibers being formed when we treated animals with SERP1 starting a week before using cardiotoxin to injure versus sham treated or saline injected animals. But in general, there's no really big differences here in terms of timing. It all went off as expected. We didn't change or improve the size of fibers, the length of time it took to make fibers. And so then we asked, well, what about in dystrophic muscles? So we used this MDX utrophin null double knockout animal. The problem with the MDX mouse is it is a disease that is geriatric, whereas in humans, muscular dystrophy is a disease of children. And it, as I said, that early onset by age 3. And the reason is that mice continue to make a protein known as utrophin. We also make it embryonically, and as we're born, it starts to turn off. So we no longer have utrophin around to help protect those muscles, whereas the MDX mouse does. So if we use this double knockout, we see onset of symptoms at around 4 weeks of age, and most of these animals will die somewhere around 12 to 15 weeks of age with increasing amounts of muscle loss, loss of ambulation, you get kyphotic spines, and they do have the characteristic defects of the heart that we see in humans. And so we started injecting these animals with SERP1 or saline, beginning at, I believe it was 4 weeks. And we continued it for 27 days. And we looked at the muscle in the diaphragms over this time frame. So we took samples after 27 days. And what we found was that we have this slight right shift that turned out to be statistically significant in those that were treated with SERP1. And what happened was that we saw increased fiber diameter when we looked at that. These animals also did not, the sham controls, we had to stop this, we were going to go longer, but the controls all started dying on us. So we had to stop it at 27 days so we could have a same age experiment. These are litter mates as often as possible. And so we then looked at all this muscle histologically. So here's wild type muscle, just wild type muscle cut. And this is a trichrome stain, and you can see there's a little bit of collagenate material, which is not shocking. Here is our untreated, oh, my wild type moved. All right, well, my label moved, it should be over there. This is the double knockout, untreated or sham treated, I should say. And you can see there's a lot of necrotic fibers here. There's a lot more collagen. There's spaces within the muscle. Whereas the animals that we treated for 27 days with SERP1, we do still see that there is some fibrotic material here, but we see bigger fibers. We don't see as many necrotic fibers, and there's one. There's another one up there. So we decrease the amount of fibrosis based on area in the diaphragm itself with the treatment with the serpin. When we looked at some of these markers of inflammation, you can see that there was much less iNOS in our PEGs, SERP treated versus saline treated muscle. And when we looked at wild type animals who were just ambulatory, we could see, a certain amount of fibrotic material. In our saline treated animals, we see much more of the muscle on any given section is fibrotic in appearance. And SERP1 is somewhat more than wild type, but certainly significantly less than our saline treated animals. And so what we're trying to do now is, oh, hang on, I got a chat, oh, I don't know what that's about. Okay, ask questions about how long can we treat for, what dosage can we use, and can we maintain these animals for a longer period of time that are getting the SERP1. So what we found so far is that SERP1 treatment decreases infiltration of macrophages, and I didn't show you that data, it does not improve acute muscle injury regeneration, and it decreases fibrosis in DMD muscle. And that was mostly the work in our lab of Alex Andre. So I'd like to thank all of my collaborators here. I have many. Kenro Kasumi and his students Cindy Hsu and Elizabeth Hutchins, Alan Rawls' student Alec and his student Alex Andre, Dale DiNardo and Sarah Stabenfeld here in SOLS and in engineering, Alex Lucas and her postdoc Li Cheng Zhang, Josh Ho and his student George Jordovic. Josh is now, they were at Victor Chang. Josh has recently moved to Hong Kong University. Tom Rando and Stefano Baresi collaborated on the numb knockouts, and Rebecca Fisher and Ron Allen collaborated on both the numb knockouts and the work on the lizards. And with that, I'm happy to take any questions.
Hello, colleagues. Are there any questions for our speaker?
So we've got one, Jeff, here in Kansas City.
Hi, Ms. Rawlson. I just had a question. You say num deficient. This goes back to the num plus and minus. Does num act on the actin and myosin of the muscle?
You know, it's really interesting question. Num actually is considered a sulfate determinant, and its job is to drag proteins together. And then in some cases, that means that the protein that it pulls along is an E3 ubiquitin ligase. And so the target will get ubiquitinated and then sent to the proteasome. And it does tend to move along actin, but it doesn't appear to act on actin. Its other jobs are a little less clear. It binds to things like P53 and MDM2 and seems to be involved in regulating some transcription factors, but the exact method, like are they being ubiquitinated, is not clear at this point.
Go ahead, Ken.
Thank you very much, Dr. Wilson-Rawls. Appreciate that. I had a hard time keeping up with all the designations of the indicators and everything, but it was really fascinating stuff. So what would the clinical application of this research be? Would it be like the prevention of myocardial hypertrophy, or what kind of clinical implications would this research imply?
So I think you're talking about the SERP work.
Mm-hmm.
Yeah, so what we're hoping, we're having the hearts from all of these animals processed right now. And what we're hoping to find is that we're going to see in Duchenne's muscular dystrophy, kids obviously get fibrotic hearts, and that's usually what they die from is heart failure, right? So yeah, we're hoping to see that. We definitely see improvement in ambulation of these mice. I was trying to get the videos. I have some videos where we've just put the animals over time in a cage and just videotape their movement, and you can just see the ones that are sham treated stop moving, they're hunched, and our treated animals look not normal, but not far off of normal. So we're hoping the hearts look better too. But yeah, that's really the hope, right? If you can limit inflammation, that would be great. I mean, these kids have to go on and off corticosteroids because you can't leave them on all the time, because I guess it also damages the heart. That's kind of like that sort of catch-22, right, of anti-inflammatories.
Thank you. Thank you very much. Great presentation.
Oh, thank you so much.
Since you mentioned ubiquitin E3 ligase, I'm duty bound to ask if simulation is also involved in that pathway.
You know, I haven't looked at that, but I have heard someone give a talk at a meeting recently, and I am drawing a blank on who it was. And it wouldn't shock you to find that out, right? Like that simulation would also be involved.
Kind of an area of interest for me right now, so thank you.
Well, you know, we should talk.
Are you a simulator, Dr. Staudinger?
Yeah, I have a lot of sumo tools in my toolkit. Dr. Ogbas, I see you were beginning to ask a question in the chat. Would you like to ask a question?
It looks like he says he had to leave.
Ah, he got hungry.
Fair.
Students, would you like?
Hi, Jeff. This is Bob White. I have a question. So I know you said the animals fared better, they looked better, they're more ambulation. How much longer did they live than that normal, like 16 weeks of life?
Well, we didn't get to find out because our saline treated animals started dying on us. So in order to have sort of comparative treatment times, we had to kill our experiment at 27 days. The vets told us the sick animals were too sick. We could no longer continue. So it would be nice to know how long you could keep them going and what would happen long term. And maybe we'll do that in the future, but we have to think of how we're going to do it because we can't do it with a control group of untreated animals.
Thank you. We got another question for you, Dr. Wilson-Rawls.
Yeah.
Hi, Dr. Wilson-Rawls. It's me again. Sorry. You mentioned lizard satellite cells forming the cartilage, and chondrogenic cells. Do those chondrogenic cells play a role on the chondrocytes or the chondrocytes really?
Well, we would like to know that. In fact, I was saying that there's a new technique out that this company has that we're trying to get them to do is where you can take a section that's paraffin embedded and then you can create a series of, select a series of sites and they will do transcriptomes up to 12 on a single section. And so what would be really great is to be able to go in early and then again a little bit later during that tail regeneration and create some sections and ask where are those cells and what do they become? So it would be really nice to know, like, do they also express PAX7 and SOX9? Like, are they somehow multifunctional cells or are we really, or are they just less regulated, right? So there's a bunch of mesenchymal stem cells and some of them are for muscle and some of them are for cartilage. It's a little hard to know. It was really interesting to us that I think it's the group that works on lizards where they transplanted GFP cells and amputated, or sort of salamanders, and amputated below those cells and then looked to see what was green. And they found that muscle really only made muscle and that cartilage really only made cartilage and bone, and that the dermis could make both bone, I believe, and dermis. So there was really this, it looked like dedifferentiation, and the whole theory was that it would become any cell, like the cells would dedifferentiate, form the blastema, they could become any cell, and then they would redifferentiate into those many cells. But it doesn't seem that way. It seems that the blastema may be full of lineage restricted stem cells, or at minimum multipotent, some multipotent and some monopotent restricted stem cells, instead of this idea of the blastema cells going all the way back to more of an ES cell-like state. So we would really like to know that too. We're trying to find ways to figure that out. Because you can't just do, immunofluorescence, because you have to do a lot of work to make an antibody recognize lizard proteins. And we found a few that do. Myosin heavy chain is one, PAX7 is another, but a lot of the ones that we've tried have not worked.
So if I understand it, a true omnipotent stem cell goes down the lineage a little ways, but chondrogenesis and myogenesis share a certain number of common pathways. And then at some point, they go one way or the other, depending on environmental cues, whether the lizard lost their tail or exercise or so on and so forth. Am I understanding that correctly?
Well, embryonically, that's absolutely correct. Embryonically, that they're all right. They're all derived from the mesoderm, right? So lateral plate versus splanchnic. So all of the musculoskeletal system is derived from splanchnic mesoderm as opposed to lateral plate mesoderm, right? You remember that.
So in a injury model, such as your lizards, are
Where,
they poised, ready to go one way or the other?
well, right. So the dogma of regeneration is that you lose that tail and then all those differentiated tissues de-differentiate, creating an ES cell-like population of cells. What we found, and what others have found, is that maybe not so much an ES cell-like population, but maybe something that reverts to a more recent stem cell. Something that in the muscle case is probably a satellite cell, but maybe a satellite cell that can also become cartilage and tendon. So maybe it's more of an embryonic cell and less of an adult satellite cell, perhaps, when you're comparing to mammals.
So then an idea might be that you could deliver therapeutic molecules that would drive it one direction or another in people, if you could understand the differences between those two fates using, I don't know, mRNA stabilized by, vaccine like mRNA stabilization technology, right?
Well, one of the things we've thought about is trying to use collagen-based scaffolds or there's some other, usable scaffolds out there that I work a lot. Sarah Stabenfeld is an engineer, and I work with her a lot on this stuff, is trying to functionalize a scaffold, use it to replace, like you could implant with satellite cells derived from a person. And could you really make this work? We can't just inject the cells. That doesn't work. They all die. They don't get to the right spot. The needle actually increases the amount of fibrosis if you're looking at muscular dystrophy muscle. People have tried that a lot, just trying to deliver satellite cells at a high dose, and they just don't implant. Tom Mirando's lab has put out a couple of papers recently where they've created a divot in the TA muscle, filled it in with a type of self-gelling gel impregnated with satellite cells, and they can get new fibers to form. The problem as it sits is that the new fibers don't actually get innervated, so they don't contract. So you can see that from an engineering standpoint, there's a lot of things that have to happen here. We're really interested in why this animal why do its cells allow it to make whole new muscles? Whereas if you and I have an injury that knocks out 80% of our bicep, we would have a useless arm, right? It would our cells would repair the remaining material, but they wouldn't remake that missing 80%. So I think we still don't know is officially the answer, but I think we have hints. And the hints are that those satellite cells are probably less lineage restricted. They're less likely to just make muscles, so maybe they can respond to a greater number of cues. If you could figure out the right cues, maybe you could then translate those cues to humans and make those satellite cells behave more like lizard satellite cells.
I get it. Some kind of ECM or positional cues that are lacking.
Well, thank you all for your attention today, and thank you again, Jeff, for inviting me.
Oh, thank you. Thank you.